Good morning, everyone. I'm Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C. Government College, Ajmer. बच्चों, मेरे लास्ट के लेक्चर में मैंने बीएससी पार्ट 2 के पेपर थर्ड के अंदर मैंने आप लोगों को एनिमल सेल एंड टिश्यू कल्चर के बारे में बताया था, जिसमें मैंने आपको प्योर कल्चर्स, डिफरेंट टाइप्स ऑफ मीडियाज़ के बारे में बताया। आज के लेक्चर में मैं आप लोगों को सेल और एनिमल कल्चर के बारे में बताने जा रही हूँ। बैक्टीरिया एंड यीस्ट कैन बी ग्रोन इन डीप सस्पेंशन कल्चर्स। बैक्टीरिया और यीस्ट जो होते हैं वो डीप सस्पेंशन कल्चर में ग्रो करते हैं, जबकि मेमेलियन सेल कैन ओनली बी ग्रोन इन इंडस्ट्रियल स्केल कल्चर्स। मेमेलियन सेल या स्तनी की कोशिकाओं को हम किस में ग्रो करते हैं? इंडस्ट्रियल स्केल कल्चर्स के अंदर। और इंडस्ट्रियल स्केल कल्चर्स के अंदर हम क्यों ग्रो करते हैं? बिकॉज द मेमेलियन सेल रिक्वायर्स अटैचमेंट टू अ suitable surface. The maximum cell numbers, however, are limited by the surface area available. The limitations can now be overcome by growing mammalian cell cultures on microcarriers. What are microcarriers? Such as beds of anion exchange resins to protect freely suspended animal cell in culture from agitation and aeration damage. Various type of additives have been used. These include, number one, these additive includes cellulose and starches. Number two, protein mixtures. Number three, dextrains, and more recently PEG, polyethylene glycol. Sixth one is the additives that protects freely suspended cells from fluid mechanical injury must decrease the frigidity of the cells. Serum permits better cell growth in agitated or aerated culture. Low serum or serum free cultures are more susceptible to fluid mechanical damage. Concentration up to 10% of fetal bovine serum FBS, it tends to reduce the cell death. Since long derivatized celluloses have been used for suspension cell culture technology, methyl cellulose MCs have not been established as reliable protectants. MCs and other derivatized celluloses have been frequently included as media additives in the cultivation of a large variety of cells. In addition to serum, several protein mixtures and a protein have been used as shear protectants for the cultivation of various cells. Again, their protective effect is not definitely established. Bovine serum albumin BSA is a major component of bovine serum, which is widely used additive in the serum free media and has been frequently used as a shear protectant, and it may be an effective additive against fluid mechanical damage of the hybridoma cells. For culture of animal cells, serum has been used as an essential component of the culture media since a long time. It may be possible that serum supplies hormones to the cell. What does the serum do? The serum supplies the hormones to the cell, and serum can be replaced by complexes of hormone, और इस सीरम को हम हार्मोन से भी रिप्लेस कर सकते हैं. फॉर एग्जाम्पल, सीरम इन मीडिया used for the culture of GH3 cells can be replaced with a mixture of insulin of the pancreas, triiodothyroxine of the thyroid, parathyroid hormone from the parathyroid, and thyroid stimulating hormone releasing hormone. Animal cell and organs can be grown by using serum as the sole support of the culture. Serum is a highly complex mixture of a diverse molecules that together provide growth promoting and growth inhibiting factors to the cell. Some of the most useful constituents of the serum include: number 1, hormones; number 2, binding proteins; number 3, attachment factors; number 4, growth factors; and fifth one is the micronutrients. Peptide hormones can produce marked proliferative effects on cell culture. Serum also contains some substances that exert protective effects on cell against harmful ingredients. Some people have used fetal bovine serum as a supplement of choice for their culture media for growing mammalian cells. Horse serum. Horse serum can be used for the support of hybridomas and parent myeloma strains and for mycoplasma. Bovine calf serum can sometimes substitute for fetal bovine serum. Porcine serum from pig, it can be used as a growth supplement for culturing human cell lines. The assemblage of cells within a tissue interact as a primary determinants of the regulation of growth and differentiation in all metazoan animals. For example, cellular interaction between the epithelium and mesenchymally derived cells. This kind of interaction is in fact ubiquitous in all tissue of higher animals. This system represents some of the hypothetical classes of tissues which are listed below. Number 1, proliferative tissue. For example, bone marrow and skin. In these, committed stem cell proliferate so long as they are attached to some extracellular matrix known as basement membrane. Another one is the differentiative tissues. For example, pancreas and endocrine cells. In these, a differentiated epithelium is bound to basement membrane, and in turn it is associated with fibroblast or endothelium. Next one is the regenerative tissue. Regenerative tissue, for example, liver, yकृत. In these, the epithelium is bound to basement membrane and it is usually associated with endothelium. Some important products procured from animal cell lines are shown in the table. As you can see that cell lines, tumor and humans, angiogenic factor, kidney and cow. The product is foot and mouth disease vaccine. Leukocytes in human, the products are interferons. Kidney in humans, urokinase. Duck embryo and fluid vaccines for rabies and rubella. Chick embryo fluid vaccine for influenza, measles and mumps. This is all about my today's lecture. Thank you.